Good afternoon, everybody. I am delighted to be here and have a chance to talk to you about an issue which indeed is very important in terms of healthcare and the community, and that has to be with, I will say C. difficile. I'll try and say difficile, but it won't come out that way, I'm sure. So we're going to talk about epidemiology. We're going to talk about some of the professional guidelines that have been published recently. And we'll talk about laboratory diagnosis because, in fact, they all are in a state of flux. Now, back in the mid-1980s, when I was the microbiology laboratory director of a hospital in Seattle, I also served on the infection control committee. And two of the critical issues we had to deal with in that time were MRSA and C. difficile. And about 5 months ago, I went back. I was invited back to give a presentation. And I talked to some of my own colleagues in infection control, and I said, well, what are the real problems you're facing? They said, well, we're worried about carbapenemase producers, but we spend most of our time with MRSA and C. difficile. So in some ways, not a lot has changed. So what we're going to do today, talk about several issues. First of all, in many hospitals, truly around the world, C. difficile infections continue unabated, and we have a variety of outbreaks, some of which are very difficult to control. There certainly are new, more aggressive, and more virulent strains of C. difficile that are emerging not only in hospitals and in healthcare, but also in community settings. Furthermore, we've got guidelines from the Society of Healthcare Epidemiologists of America and the Infectious Disease Society of America. They recommend that enzyme immunoassay tests for toxin A and B not be used for routine testing. In fact, they advocate using a GDH-based algorithm, but they say you've got to confirm these with one of two reference methods. You've got to either use toxigenic culture or direct cytotoxin testing for confirmation. And unfortunately, not many laboratories actually use those reference methods. So the question that comes up is, what should you be using for diagnosis of C. difficile if you're going to try and control your outbreaks? And why do you need to pay attention to this? So, the organism, as many of you know, gram-positive anaerobic spore-forming bacillus, and the fact that it forms spores is really critical to its epidemiology. Those spores are very hardy. They hang around in a hospital for a long period of time, and in fact, to get rid of them, many hospitals have found they have to go to bleach. The other issue that's come up a lot is that we've spent in infection control the last decade trying to convince hospital staff that they should use alcohol-based hand rubs in order to reduce infection rates. And that's absolutely true. And they work very, very well, except for C. difficile. I mean, in the laboratory, how do we enrich for C. difficile? We put the stool in alcohol. The same thing we're putting on our hands. And so those people who've undergone outbreaks in their hospital have said, well, you know, we need to do something else. We need to tell people, and this is very difficult, to go back to soap and water. Why? Not because soap kills the organism, it's not going to kill the spores, but the flushing action gets it off your hands. And we know that hands are certainly one way that we can transmit this infection around the hospital. So in terms of infection control, sometimes that's difficult. So where have we been? Well, the Centers for Disease Control predicted, based on two types of data, that our rates were just going to be continuing to go up and up and up. And in fact, that's pretty much where we are. The rates, at least in the US, continue to go up. And why do we pay attention to this? Well, there was an article that appeared in December in the Archives of Internal Medicine where they looked at the issue of in-hospital mortality. In other words, what is the effect of having all of these C. difficile infections? And what they noted is, on average, there was a threefold increase in the hazard of death associated with a hospital-acquired Clostridium difficile infection. And in fact, if you look across all baseline strata, that is, no matter where patient age, what their risk factors were, for every 10 patients that acquired the infection, 1 patient died. So C. diff is not just a nuisance in the hospital, it's a significant cause of morbidity and mortality. And now the CDC is suggesting that in the US, based on more recent data, we may be looking at up to 3 million C. difficile infections annually with a mortality rate that's at least 15,000 and probably at least double that. Now, recent outbreaks of severe disease have been caused by the epidemic strain, which goes by at least 4 names. North American Profile 1, or NAP1, if you type by pulse field. O27, if you do ribotyping. BI, if you do restriction endonuclease analysis. And toxinotype 3, if that's what you do. It's all basically the same organism. But it does have a propensity to cause epidemics, and it certainly is more virulent than most strains, and it tends to be fluoroquinolone resistant. So if you're in your hospital or in your community, if you use a lot of fluoroquinolones, like levofloxacin, like Cipro, then you are at increased risk for having this strain certainly develop and emerge. And I know there's hospitals in the United States that use so much levofloxacin, it's affectionately referred to by the pharmacy as vitamin L. Now, the elderly, clearly, are the most common group of people that get C. difficile. But in fact, we're starting to see it emerge in a lot of low-risk people, including very healthy people in the community, including people that have had absolutely no contact with the healthcare system. So we've always thought about this as a nosocomial infection, but we're really going to have to start changing our minds. Oops. And then the last point is, and I think this is truly something we need to pay attention to. C. difficile has been isolated from retail meats in about 20 different countries. It's been isolated from beef, from pork, from turkey, and in Scotland we know it was even isolated from readily prepared salads. So go to your favorite deli, buy a salad, oops, C. difficile. Now, that doesn't mean that we've confirmed that this is a foodborne infection. As a matter of fact, there is no smoking gun. There is no outbreak of C. difficile that we can trace directly back to food. Yet. So we'll see. Now, when you look at foodborne outbreaks, and this was an article in Clinical Infectious Diseases that appeared last year from the CDC, they were asking whether this is in fact a new foodborne pathogen. And they looked at all of the various articles that had reported the recovery of C. difficile from a variety of meats in the United States, Canada, and Scotland. And in fact, they also had a variety of publications from the Netherlands, from Belgium, from Germany. And what they noticed was that a lot of these were either PCR Ribotype 027 or 078. And 078 has always been associated with animals. As a matter of fact, there was a time when it was thought to be exclusively a C. difficile strain of animals, but now it has crossed over and we're seeing a variety of human infections with 078 as well. There are a variety of other types that you can see, but these are the ones that really jump out at you. Which means if we're going to understand the epidemiology of C. difficile, we really need to know something about strain typing. Now, in this study, which was just accepted for publication in the Journal of Clinical Micro, we've looked at PCR ribotypes, we looked at REA typing, and we looked at pulse field. And basically, we see that there's very good concordance among these 3 methods for calling 027BI NAP1 strains. So everything looked pretty good. There weren't any strains that were just PCR ribotype 027 and not these other types. Only one that was just BI and not the others, and only 2 that were just NAP1. So there is pretty good concordance for that. However, when you move out and look at other PCR ribotypes compared to REA and pulse field, then we see things aren't so clear. So if you look at all of the type Ys here, they represent a variety of different ribotypes. So does type BK. And you can see down here that type 104 and type 106 actually share the same pulse field profile. So it gets a little bit murky when we're really trying to understand the epidemiology of C. difficile. But fortunately, this clears it all up for you. Right? Well, anyways, this is an attempt. This is looking just at 078 strains. And it's actually simpler than it looks because everything here in green, even including the pink, those are all highly related O78 strains, greater than 85% related. And in here you can find both human and animal strains. And so again, we're starting to make some links due to typing that we couldn't make before. Now, one of the interesting points of this study, which had multiple co-workers from multiple countries, is they looked at antimicrobial resistance. And one of the things they noted was that tetracycline resistance was seen in O78 strains, and it was all from TN916. So TN916 is a very common transposon mediating tetracycline resistance, usually found in gram-positives like enterococci, but it's also been found in pneumococci, and now it's showing up in Clostridium difficile, which means that there's probably some commonality and sharing of resistance genes, and we never thought about that before. And think about something else. What's one of the most common resistance genes we see in enterococci that's transferable? Vancomycin resistance. What do these data show? These data show that there's absolutely no reason why we should not be seeing high-level vancomycin resistance in C. difficile in the future. Clearly, the pathway for transmission is there. So, the O27 strain, most of you are probably familiar with this, but it's probably good just to go back and revisit the epidemiology. Historically, this is a very uncommon strain. So if you go back to strain collections like Dr. Dale Gerding's in the US, which goes all the way back to the early 1970s, and you look for this strain, it is rare, very uncommon. And when you find it, it's almost always, in fact, it's always fluoroquinolone susceptible. So this is a strain that has clearly evolved over time. It's now epidemic in North America. At last look nationally, about 32% of all C. diff strains were O27. It's clearly in Europe, and now it's emerged in Australia, which was thought up until a year ago to be O27 free. Carries an extra toxin called the binary toxin, which adds to the pathogenicity of this strain. It's sort of interesting that by itself doesn't seem to cause disease, but put it with toxin A and B, and it really seems to enhance the virulence of the organism. And it has an interesting genetic defect, a polymorphism in the control locus TCD-C that seems to upregulate toxin A and B. These strains just make a lot more of that toxin. And we presume that that has something to do with the virulence, that if you had more toxin production, you're probably going to be more virulent. But this is an in vitro phenomenon. We have not proved that yet in vivo. And it makes a ton of spores, which probably contributes to its causing epidemics because it makes so many spores and it's very hard to get rid of in the hospital setting. Now, this is an article that appeared in the New England Journal of Medicine about 3 weeks ago, so hot off the press, if you will. It looks at fidaxomicin, which is a new drug. It's a macrocyclic drug, first in class. It was compared to vancomycin for C. difficile infections, and what you can see is if you look at the clinical cure, there's really no statistical difference between fidaxomicin and vancomycin. Vancomycin being the drug that's recommended for severe infections. Now, if you look at global cure, you see there actually is a significant difference. Now, what's the difference between clinical and global? Well, it's a little hard to figure out of the paper, but these people feel better, they look better, but they may still have a little bit of diarrhea left around. But when you go for global cure, which means these people, they're stopped, then fidaxomicin's a little bit better. But what is important is that fidaxomicin has fewer recurrences. That's very important because a lot of C. difficile patients will get recurrent disease. And if you get one recurrence, that means you're at risk to get more recurrences. Now, something else that was in the article but sort of buried is shown in this slide from a poster from ICAC, and that is when you look at cure rates for vancomycin versus fidaxomicin, O27 strains are statistically significantly lower for both drugs than any other type that you look at. So these are very good drugs except for this. So patients with O27, first of all, are harder to treat no matter what drug you're given, and they're more likely to relapse. This is an article from Tom Riley and colleagues looking at O27s around the world. And gray does not mean no O27. Gray means lack of data. We don't really know what's going on. And I was in Australia 3 weeks ago, and I learned that O27 strains are not just now in Western Australia. They've been found in Brisbane up here in Queensland. They've also been found in Sydney and in Melbourne. So O27 has basically filled in the map of Australia now, and they're very, very worried about the spread of those strains. Interestingly enough, there are O27 strains in Japan, and they're all fluoroquinolone susceptible. We're not quite sure why that is. Another interesting thing about Australia is those strains are all fluoroquinolone resistant, even though they use very, very little fluoroquinolones in Australia. They're tightly, tightly controlled drugs. And I think you can probably just color in all of the states here in the U.S. because, as you see, it's basically north to south, east to west. We have O27 anywhere. And of course, in Europe, there are a number of outbreaks that have been attributed to the O27 strain. Now, in terms of the epidemiology, this was a very interesting article that appeared a couple of years ago. It was from the state of North Carolina, where they did over 1,000 chart reviews of patients who had acquired C. difficile. And one of the things they noted was that 44% of the cases had community onset disease. Now, that's actually not so surprising because length of stay in the United States is usually pretty short. People come in, they get tuned up, and they go out the door. And in many cases, people come in, they get tuned up, they acquire C. difficile, then they go home, and then they have their disease out of the hospital. But what really was fascinating was when they went back and looked at those, 20% of those patients that had C. difficile onset in the community had no link to health care. It was truly community-acquired disease. Now, I'm not sure about here in the UK, but in the US, a lot of laboratories would never test an outpatient stool for C. difficile. Why? Well, because it's a nosocomial pathogen, right? Well, maybe not anymore. Maybe we really need to think about the fact that this is just as much a community pathogen now as it was in the past. And my hat's off to you because, in fact, you have shown a decrease, and we hope that this is sustained over time. But I think it just shows when you put your mind to a problem, even one as big as C. diff, you certainly can have an impact. Okay, let's talk a little bit now about outbreaks and how to get rid of them. And clearly, outbreak investigations need to be conducted by highly trained individuals, and maybe not by this guy who obviously missed his class on personal protective equipment that particular day. Okay, there was an interesting study published by Carlene Muto and colleagues from Pittsburgh. They had a very, very long outbreak of C. difficile in their hospital, and I'll show you that epidemic curve in a second. What they did is, through their experience, basically said, you know, you can't just do one or two things. You need a whole bundle of practices if you're going to be able to deal with an outbreak. And they had sort of their 5-component intervention that they used, that they developed over time. First one, education. Everybody from the CEO to the janitor that sweeps the floor has to understand what C. difficile is and how to prevent it. Second of all, early case finding. That is, the infection control people would immediately go and do chart reviews very early on when a patient had that third loose stool in a day. They wanted to look for risk factors. They wanted to find people. They wanted to get them into isolation early. Expanded infection control, I'll talk about that a little bit more in the next slide. Targeted antimicrobial management. Antibiotics are a huge risk factor, particularly fluoroquinolones. And so if you can control that selective pressure, then you can help get rid of this organism in your hospital. And creation of a C. difficile management team. And this actually included one of the chief medical officers was on this team. That's how much of a problem it was. And those people met basically daily during their outbreak reviews. So, infection control, enhanced environmental cleaning, using a lot of bleach. Now, people don't like to use bleach. It smells bad. It's hard on the skin. It's hard on equipment. There are certain things that you don't want to bleach, like portable X-ray machines, but you have to really pay attention to cleaning. Electronic flags on the charts. You certainly do not want to put an infected person in the room with an uninfected person. You don't want to have that transmission opportunity. Hand hygiene with soap and water. We talked a little bit about that before. These people thought that that message was important. It helped emphasize the fact that C. diff's outbreaks were different. They needed to be controlled, but alcohol hand gels worked for everything else. If you got C. diff, you basically stayed in isolation until you came out of the hospital. And then infection control audits. You have to monitor what's going on. You can't have a policy that says hand hygiene with soap and water and just leave it at that. You need to monitor people washing their hands, and when they're not doing it right, you have to tell them so. So here's the epidemic curve. Starting back here in 1999, it went up dramatically, and you can see these are all of the various interventions they were trying and get rid of this, and you see that it's only when they're getting out here it finally starts to come down. And this indeed, once again, was the 027 strain. I went back and looked at this article again because I've been using this article to talk about how bad 027 is for a while. And it just struck me that it took so long to get this outbreak under control. And I said, well, what were they using to detect their cases? And they were using direct cytotoxin testing, which in their hands probably had a sensitivity of around 70%. Now let me ask you this. How difficult is it to control an outbreak when you miss 30% of your cases? I think that could be a problem. Okay, let's talk about another outbreak. Alex Callan and colleagues at the CDC were involved in another hospital, and they had an outbreak of C. diff, and they did basically everything they did in Pittsburgh, and they were still having problems. And so what they decided was that they really had to focus on the fluoroquinolones because this hospital used a lot of fluoroquinolones. And indeed, what their study showed is that really was critical for interrupting the spread. Now, I went back and looked at the lab methods in this paper as well, and this was a little bit confusing, because they said that stools were frozen and sent to CDC for culture and strain typing. There's no mention of doing any other testing on site for C. difficile. They probably were doing something, but there's no mention of it in the paper. So here's their outbreak period, starting in January of 05 through 06, and they're out here to 007. This is where they put the fluoroquinolone restrictions in place. This gives you a sense about how difficult this is to do because they had an absolute ban on fluoroquinolones, and yet the actual fluoroquinolone decrease was only 66%. So a third of the time, even with an absolute ban, and this is partly because they had a lot of GPs and other people, other physicians that admitted to the hospital, and they could control what went on in the hospital, but not what went on in the community. So people were coming in often on fluoroquinolones, and they had to sort of step these people down or put them on something else. Well, they finally got a fluoroquinolone restriction in place. They got a 22% decrease in C. difficile infections. They also noted that they changed their contractors for their janitors in the hospital about the same time. Not sure if that had anything to do with it, but once again, an intervention seemed to work, and then things are drifting back down, and here they actually added the fluoroquinolones back again, but at a certain reduced level. But certainly, they attributed this block to their fluoroquinolone restrictions. And what about the spores? Well, this was an interesting article. What's on that keyboard detecting the hidden environmental reservoirs of C. diff, particularly 027? Now, what these folks did is they did a lot of cultures around the hospital, over 100 cultures. And what they found was 16% of those were contaminated. They were positive and grew out toxigenic C. difficile. 31% of physician work areas, that is non-patient care areas, but only 10% of nurse work areas. No comment on that one. But this is what I found interesting. 21% of portable equipment that you bring into the rooms were culture positive for C. difficile. Now, this is something that I think a lot of people had not paid attention to before. So you might have an immaculately clean room, bring in that portable X-ray machine, and boom, now it's contaminated. 50% of all the strains they isolated were, in fact, the NAP1 epidemic strain. Okay, let's talk a little bit about laboratory now. These are the guidelines from the Shea IDSA that were published at the end of last year. I'm just going to give you 3 key points, 3 snippets out of this. Enzyme immunoassays for toxin A and B, rapid but less sensitive than cell cytotoxin assay, and is considered suboptimal for diagnosis. And yet, if you're looking at surveys in the US, this still is the most commonly used laboratory method. Overall, probable sensitivity, 50%. Most of these tests were actually approved during an era when direct cytotoxin was considered the gold standard, and they were approved primarily during periods or evaluated during outbreak or in areas that had very high prevalence. They next say, basically, a potential strategy to overcome the problem of lack of sensitivity is using a 2-step method. GDH is the initial screen, and then use cell cytotoxicity or toxigenic culture as the confirmatory test. Basically, it's saying the EIA test for A and B can't be used for confirmation because they're still only 50% sensitive, even as a confirmation assay. And yet, you look around the US, very, very few laboratories do this, the direct cytotoxin test, partly because it takes 48 hours, but mostly because many laboratories don't have cell lines around the way they used to. And toxigenic culture, once again, anaerobic cultures, something that's, except for reference laboratories, has really gone the way of a number of other tests. So, what did the guidelines say about PCR? PCR testing appears to be rapid, sensitive, and specific, and may ultimately address testing concerns, but we need more data. And I think when this was published, that was a fair statement, because this guideline was actually written and completed almost a year before it was published, and at that time there weren't a lot of articles about PCR. But now, I think we have to answer this question. What does the published evidence say now about PCR and GDH as routine methods for C. difficile? All right, well, let's take a look. Let's see what it says. This is an article from the Mayo Clinic from Lynn Sloan and colleagues. And what they showed was now using broth-enriched toxigenic culture as the gold standard, the 3 most common EIA toxin tests used in the US, less than 50% sensitive. You might as well flip a coin. The GDH test, rapid GDH test, 76% sensitivity. Now, look at this, because we'll come back to this. I thought this was intriguing. This is the Mayo Clinic. Their homebrew PCR had a sensitivity of 86% when compared to broth-enriched toxigenic culture. Here's an article that appeared last year from Susan Novak Weekly. She manages a very large reference laboratory in California, and they looked at the impact of a variety of different algorithms, but really focused in on how does GDH help you or hurt you. And so when they looked at EIA alone, this is EIA for toxin A and B, sensitivity was 58%. When they added GDH as a screen first, sensitivity actually dropped to around 55%. Probably not statistically significant, but it was a drop. They ran PCR, the gene Xpert, sensitivity in their hands, 94.4%. Used the GDH as a screen first, 86% sensitivity. Use GDH with EIA, and if EIA is negative, then do direct cytotoxin, because in fact this was an algorithm that they were already using in the lab, right in the middle, about 83%. So one of the things that these investigators concluded from their study was that GDH actually reduced the sensitivity of both EIA and PCR testing. And they, like many of us who've been teaching for many years said, well, you know, GDH is 100% sensitive, right? Every C. diff has it, and even some Clostridia that aren't C. difficile have it. But these data from this recent study, and once again, gold standard was broth-enriched toxigenic culture, suggested that maybe it wasn't quite as sensitive. Here's a recent review from Cepheid and colleagues, published in the Journal of Hospital Infection, looking at a meta-analysis of GDH. Now, one of the things that struck me about this is, right, GDH is supposed to be 100% sensitive. And in fact, you look down sensitivity and there's only one study that actually found it 100% sensitive. Now, these are compared either to direct cytotoxin or toxigenic culture. And let's say broth-enriched toxigenic culture is the gold standard you're really interested in. Then what's the sensitivity of GDH screening? Well, 87% here with a prevalence of 17.5%. Prevalence of less than 10%, 8.6. Sensitivity 95. That's pretty good. Here, prevalence of basically 22%, sensitivity of 80%. So what we're seeing now when we look at a better gold standard, if you will, just like we've been looking at the EIA, is those GDH assays aren't nearly as sensitive as we thought they were. You may take anywhere from a 10 to a 20% hit in sensitivity using it as a screen. The American Society for Microbiology published some guidelines as well. Highlights from those are testing, limit that to symptomatic patients. Those patients who have at least 3 non-formed stools in a 24-hour period. So if the stool comes down to the lab and it rattles in the can, no testing for C. diff. Have to be symptomatic. Toxin A and B, too insensitive, don't use them. Glutamate dehydrogenase, good screening test, but follow it up with a confirmation assay. Once again, sensitivity, high, specificity, low because it detects non-toxigenic strains as well as some strains that aren't even C. diff. Fifth point, PCR can be used to detect C. diff or C. difficile toxin genes as a standalone diagnostic test. So to make a long document pretty short, they end up with 3 algorithms. Use a standalone GDH assay, you get a positive. Use a toxin A and B if you want, or direct cytotoxin. If they're both positive, stop. If it's negative, you have to do something else. Use either a PCR assay or toxigenic culture before you're done. And there have been several nice publications, including one out of Barrow's laboratory, that basically follow this same approach. You can use a GDH-EIA combination like you can find in a lateral flow assay. Both positive, stop. Both negative, stop. GDH positive, EIA negative, check that negative. PCR or toxigenic culture. But as Earl already told you in the US, that basically means PCR. Or ASM says PCR is the standalone test, most sensitive and specific. PCR positive, stop. PCR negative, stop. So you've got basically 2 approaches, one with GDH and one with PCR. So what is the gold standard for testing? Well, in the literature, there's 3. There's toxigenic culture, there's broth-enriched toxigenic culture, and there's direct cytotoxin testing. All of those are considered as valid gold standards. However, none of these tests are commonly used in most clinical laboratories. How many people, anybody here do direct cytotoxin testing as their usual assay? Either no or you're all shy. Okay. So, if you look at the direct cytotoxin literature now, not GDH, but direct cytotoxin, which I haven't shown you, basically the bottom line, and this was published in 2 reviews by Lance Peterson recently, direct cytotoxin testing is somewhere around 70, perhaps 80% sensitive. Okay? Now, some would argue that this is the best indicator of disease because that is the toxin, that's the major virulence factor. So you're finding the major virulence factor expressed in the stool. Incontrovertible proof, right, that that patient has disease. No argument there. here's my quibble. How can you have a gold standard that misses 30% of true positives? Is that really a gold standard? We can have an interesting discussion about that. Okay, there are, in fact, 4 molecular amplification tests for C. diff. The first one out in the market, or at least in the US, was the BD genome. Very good test. Targets TCDB, that's the toxin B gene. It's batch-oriented. Get your results in 75 to 90 minutes. There's the Prodessa Progastro. This is a little different approach. You have to do a DNA isolation step first, then you put the isolated DNA into the test. So that one takes you about 3 hours. Again, targets toxin B. The Cepheid gene Xpert, on-demand testing, targets TCDB, as well as tells you whether it's O27 or not. That can be done in 45 minutes. And then the newest entry is the Meridian Illumigene. This is not PCR. This is loop-mediated isothermal amplification, otherwise known as LAMP. Yeah, I practiced that, so I get it right. So, it targets TCDA. Now, there are TCDA, toxin A negative strains, but most of those have smaller deletions or deletions at the 3' end, and these amplification primers are at the 5' conserved end, and so we're assuming that these actually won't miss those like Ribotype 17 strains, and you get your results in about 1 hour. Now, if you look at the package inserts, these are the number of steps it takes. So anything that's stippled like this means that's an incubation, that's hands-off. So Gene Xpert, as you know, swab into stool, stool into buffer, buffer into cartridge, cartridge into the machine, and you're done. Prodessa, remember, you have to isolate DNA first, so there's lots of steps. The Illumigene has 7 steps here with some incubations in between, and the BD genome has multiple steps. And here's basically the amount of time it takes you to get your answer. So hands-on time is a little bit different, varies from test to test. So let's look at a little bit of published literature. Once again, focusing on those studies in which broth-enriched toxigenic culture was the gold standard. This is one from Johns Hopkins University from Karen Carroll, who's a well-known microbiologist in the US, looking at the progastro, sensitivity of only around 77%. Actually remarkably low for a PCR test. Using that same gold standard, same laboratory, looking at the BD genome test, around 84% sensitive. Again, a little bit more, a little lower than I would have anticipated, but pretty much in line with that 86% of the homebrew assay from the Mayo Clinic. This is the clinical trials of the Gene Xpert. Overall sensitivity against compared with a variety of methods was 93.5% using broth enrichment. Now, if you just did direct plating of the stool on CCFA agar, that sensitivity would have been 98.8%. And I show that just to help you understand that that broth enrichment part of toxigenic culture really does make a difference. So this drops down because you're finding a lot more positive results when you do that broth enrichment. But basically, overall, again, around 94% sensitive, very similar to what Susan Novak and colleagues found. So I have a hard time putting all of this together, so I thought I would do a chart. And so what we've done, and I did this in collaboration with Lance Peterson from Northwestern Hospital, was looking at C. difficile infection detection. And this is the model that we sort of built. Let's take 1,000 consecutive patients. And in fact, this model was just accepted for publication in the American Journal of Medicine, will be out in about 2 months. But just based on literature results, we decided to prospectively test 1,000 patients, and we're going to assume the prevalence of CDI is 10%. So we've got 100 true positives and 900 true negatives. And looking at about 4 publications, GDH, EIA together, we gave a sensitivity of 55%. It varies from 50 to about 72%. But we're also giving GDH the benefit of the doubt based on 2 publications, including Susan Novak Weekly's. So GDH alone has a sensitivity of 87%. And in the US, average length of stay, 5 days. Okay, so what are we looking at here? Well, we're looking at if you do GDH and EIA alone, even if it's GDH positive, EIA negative, and you just stop there, these are average costs for a number of tests now in the US, versus if you get the EIA negative reflexing to PCR, reflexing to toxigenic culture, or reflexing to direct cytotoxin, and then comparing those results with PCR alone. So this is actually an average of looking at BD, Cepheid, and Progastra in terms of their costs. So that may look a little bit low compared to what you pay, but remember that's an average of all of those tests together. So sensitivity 55%. If you take those EIA negatives and you reflex to PCR, that's going to go up to 83%. If you look at PCR alone, 95%. This is based on multiple assays, and I'll show you that again. So basically, what's the number of patients you're going to miss? If you just do GDH EIA alone, you're going to miss 45. You do PCR alone, you're going to miss 5. If you reflex to PCR now, you're going to only miss 17. You're going to pick up a lot of those patients. How about for the specificity side? All right, well, if your specificity is 94%, which is pretty good, and that's exactly what's published for a couple of articles, that means you're going to have 60 false positive results. Compared to PCR down here, 96%, you're going to have 40 false positives. You're going to, remember, you can never get better than 87% sensitivity for any of these reflex tests. Now, what does that mean in terms of people actually going into isolation? This is sort of what I found interesting. Now remember, for toxigenic culture, it's going to take you 4 to 5 days to get that answer, which means there's 4 to 5 days that these people are not on therapy and they're actually shedding unless they're in isolation. So here you've got patients, 115 patients. You've got your 60 false positives, and you've got your 40 or your 55 true positives there. So you're going to have 115 patients in isolation for 5 days, their length of stay, 60 of which are actually don't even belong there. PCR, you're going to have 135 patients for 5 days, of which actually only 40 don't belong. And then as you start to play with these various reflex tests, you see we really do improve things. We kick out a lot of patients that don't belong there and end up with more people who actually do belong there. And so the bottom line is GDH followed by EIA followed by PCR looks like it could be a very cost-effective way to go. You don't miss a lot of patients. You get the right patients in isolation, but you still have a better deal if you go with PCR alone. But it's not a bad way to go. Now, during our clinical trials, it really bothered me. I kept reading the literature and saying, how can 3 or 4 laboratories all use the same test, the EIA, toxin A and B, and these people find 86% sensitive and these people find 48% sensitive? Is it more than just the testing? And so we said, well, what if we look at the data by ribotype? Is it going to tell us something different? And in fact, it did, and it was quite surprising. So if you look at the sensitivity of an EIA test for ribotype 002, you'll see that the Xpert was 100% sensitive. The EIA test for toxin A and B was 15% sensitive. What does that mean? It means if you have an outbreak of ribotype 002 in your hospital, you probably won't know it because all those patients are going to show up as negative. Even for 027, 100% sensitive, 78% sensitivity for EIA. And down here for ribotype 106, which is not all that uncommon in the UK, less than 20%. So depending on your strain mix, you can get vastly different results, even from people who are just across town with EIA. Now, how about GDH? This was also interesting. If you look at 027 strains, there was no difference whatsoever between GDH and GeneXpert. They were exactly the same in their sensitivity. However, if you look at ribotypes other than O27, the GDH actually drops down in sensitivity to about 70%. Once again, your strain mix can have a remarkable impact on the sensitivity of the test that you're using. So, GDH versus PCR. GDH algorithms that include PCR for the GDH positive EIE negative results have been recommended by multiple studies in the literature. I can think of at least 4 off the top of my head that say this is a cost-effective way to go, and it's one of the algorithms suggested by the American Society for Microbiology. Using PCR alone has also been recommended by ASM, as well as other published articles. So the GDH and PCR approaches have pluses and minuses, including sensitivity, but also including cost and turnaround time. How fast do you need those answers? How much money do you have to get those answers? And what hit in sensitivity are you willing to tolerate in your hospital system? The other thing that seems to be emerging from the literature, and this needs more data, but I thought it was intriguing, is that there appear to be 2 levels of sensitivity for PCR assays. You can look at the ones published by Sloan, by Liu and Benai, Stamper and colleagues, and at least 2 assays. PCR running from 77 to 87% sensitivity, including the homebrew assay by Stanford University and their molecular biology group. On the other hand, you look at Novak Weekly, you look at Wong et al., you look at Babity et al., you look at our clinical trials, and you see 93 to 100% sensitivity compared to toxigenic culture. Now, is this because it really is dependent on the ability of the assay to lyse spores? Maybe that's the difference between vegetative cells and spores. We don't know yet. Tom Riley in Australia is actually doing a study now looking at a variety of ribotypes doing pure vegetative cell preps versus total spore preps, and hopefully we'll be able to get an answer to this. So labs really have to decide which approach is best for patient care in their hospital. If you switch to PCR, when do you have to do a repeat test? Well, here's a study from Stanford University. They looked at repeat testing on almost 300 patients, and basically the bottom line was repeating a PCR test within 7 days is rarely useful. So you know you're used to EIA, toxin A and B, you get stools times 3, or sometimes times 5, or sometimes times 7, because they're negative, and the doc keeps saying, I know this patient has C. diff, send another stool. I'm going to send them till I get my positive. Okay? These data basically say, do 1 PCR test. If it's negative, 7 days later, you may think about doing another one, or if there's evidence of a new infection. But once again, homebrew PCR from Stanford, 87% sensitive. Something to think about. Now, Dale Gerding, who is an aficionado of C. difficile, been working on C. difficile for close to 4 decades, presented this slide at the 5th Decennial Conference on Healthcare-Associated Infections in Atlanta. He said, this is where C. diff rates are going to go. They're either going to go up, they're going to stay the same, they're going to go up, then they're going to go down, or they're going to go down. And he said, I don't know which one it is. It's one of those 4. And I totally agree with that. It's one of those 4. But I have an opinion. My opinion is C. And I think we're already starting to see this. And I'll tell you why. As laboratories switch to PCR, I think, to molecular methods, they find a lot more cases of C. difficile in their hospital. Their sensitivity is almost doubled. As a matter of fact, I've had at least 3 hospitals call me after they've started the Gene Xpert and said, Fred, my rates went up by 27%. Said, well, your rates of detection went up. The rates of infection didn't go up. You're just finding all those cases that you were missing by EIA. And you know what they say after that? My rates are going down. Why? Because once you start finding all those cases, your infection control actually works a lot better. And your rates will start to go down. But you can't get your rates to do this if you're missing 50% of your cases. Okay, so let's wrap up where we've been. Epidemiology of C. difficile infection is changing with more community-associated disease, and intriguingly, CDI may be a foodborne illness. We don't know yet. It's possible. Infection control for CDI is a challenge, especially when the laboratory tests miss 50% of the positive patients. The SHEA IDSA guidelines don't recommend PCR-based testing for CDI, but both they and the ASM guidelines suggest it may be the most sensitive and specific method available. However, more recent data, I think, puts PCR in a better light. Clearly, laboratories need to stop doing toxin A and B EIA tests and implement more accurate tests, either PCR or GDH algorithms with follow-up of the EIA negative test, if they're going to enhance detection of C. diffs and if they're going to control transmission. Clearly, one of those algorithms is the way that we have to go. Even if you reflex your GDH positive EIA negative to toxigenic culture or to direct cytotoxin, you're going to be better off than just stopping with GDH EIA alone. I think one of the things to watch in the future, and in fact there are several studies in process right now, is this idea of antigenic variation in GDH among C. diff strains. Because in fact that may be limiting its sensitivity, as suggested for the non-O27 strains in the clinical study we did. We clearly need some more data on this though, because I think GDH right now is an acceptable algorithm. But once again, it looks like you are going to be taking a hit in sensitivity. And with that, I will stop and thank you for your attention.